Okay, welcome. Welcome to our next Nuvisan webinar or series of webinars. Today we'll speak about bioanalysis, bioanalysis in the context of biomarker assays, and today we will focus particularly on complex analytical workflows and how they interact with bioanalytical biomarker assays. So your host today, my name is Achim Freisleben. I'm one of the managing directors in Nuvisan and head of bioanalysis. And with me today is Heike.
Yeah, hello. So my name is Heike Wiese and I'm a senior scientist working in LC-MS method development and validation.
Okay. So today's agenda. We will speak briefly about our services and service portfolio to get an understanding who Nuvisan is and where we are involved. Heike will speak briefly about biomarkers in general, and then we are going to share a couple of case examples for you, focusing on various aspects of biomarker assays. We have a selection for you, which I believe is very interesting, focusing on microsampling as a first example, speak about the RAS biomarker assay, and close off with an enzymatic assay as CONT. And of course, we will have a Q&A session at the end of the webinar. And I hope that you have a couple of questions, and we will be happy to speak about those. A few house rules for the webinar. Maybe to start, giving you an impression of the Corona policies. So we are here today standing pretty close to each other. And in order to have, as good as possible for today, a reflection of the Corona policy, we've put up this wall here. It shouldn't hurt or it shouldn't harm the picture that you are seeing. Secondly, your microphones are are muted by default. Sorry for that. In case you have questions, please take them down and note them in the Q&A chat box, which is on the lower part of your Zoom screen. And in case you don't have any questions immediately, you want to follow up with us later on, don't hesitate to get in touch with us after the webinar. You will be getting a recording and a transcription of the slides. So you have ample opportunity to get in touch with us. So who is Nuvisan? Just a very brief overview, just 2 slides, who we are and what we do in this respect. Nuvisan started off about 40 years ago as a bioanalytical CRO. At that time, it was called LAB. Along the road, we added additional services, early clinical trials, pharmaceutical services, and non-clinical DMPK services, to name but a few. Nuvisan has evolved also from a CRO into a CDMO, so we are also offering manufacturing services. Most recently in 2020, early 2020, we signed a contract with Bayer Healthcare. So Nuvisan took over a good part of the Bayer R&D facilities located in Berlin, and we named it Innovation Campus Berlin. And you see that now we are starting from the earliest phases of research throughout the early development path, offering services along the value chain. Why is that important for today? Today we're speaking about biomarker services, and the examples that Heike will be presenting later on are focused on development biomarker assays with different stages of qualification or validation. But of course, we do have in our screening hit ID cascade, we do have a biomarker research group in Berlin that offers potential to support us in all kinds of questions along with biomarker research, biomarker identification, assay selection, biomarker implementation strategies, and those could be brought together with the development capabilities in Nuvisan that we are speaking about today. Just a very quick snapshot on what we offer in bioanalysis, and I'm not going to read all of this slide out for you. It's very detailed and for your reading. I believe the important point to highlight is that we can offer all bioanalytical services under one roof, including also a clinical phase 1 unit here, located here in Neu-Ulm. And that is also important for a couple of the examples that Heike will be showing. So we have direct access to fresh blood from healthy volunteers. So we can take fresh blood and take them to our laboratories and build up the assays using authentic matrix, and we can also do cell preparations in our cell culture lab if that would be a necessity. And we also do have the small and the large molecule bioanalytical teams located here in one facility. So I believe that it's ideally suited to address the biomarker assay questions that people tend to have in nowadays projects. So I think that's very worthwhile to mention. And apart from that, we do have all the techniques and technologies that you would be expecting from us. That brings us, Heike, to the first poll.
We would like to ask you first if you ever have been working with Nuvisan before. And we would like to ask you to answer the question. For any Nuvisan employees who are listening, you don't have to answer this question. Yeah.
Just a few seconds more.
Okay. Some new faces.
Yeah, there's it's pretty mixed, the distribution that we are having, but more or less 50% of the people has never worked before with Nuvisan. That is good, of course, at the one hand, unfortunate for us on the other hand, but it's good because that's why we are here for, and in order to talk about the services and some case examples that we have put together. Heike, please.
Okay. Ich hatte die Maus hier nicht. Okay, so today we want to talk about biomarker assays, and we all know biomarkers are measurable characteristics of an organism, be it blood pressure or an MRT picture. But of course, when you want to use LC-MS, we are always talking about a molecular entity we want to measure. And important to notice is that because this is a characteristic of the organism, we are always having endogenous compounds as an analyte. So that means that we can't apply PK guidelines for validation. And now the question is, what level of validation do we need for our assays? And that's highly dependent on the stage of your drug development and the context of use. So do you have a high or low endogenous level? Do you have a big biological variance or not? Is your biomarker increasing during treatment or the disease, or is it increasing? And what do you want to do with the biomarker data? So is it just for internal decision making or data collection, or is the data meant to support decision making by the FDA, for example? So we want to talk about LC-MS, and we really like that technique because as an analytical technique, it offers high precision and robustness, and so it can be used if high precision is needed. And we also want to talk about implementing complex workflows. And what do I mean by complex workflows? So a standard method might be to draw blood and then extract your analyte from plasma and measure it by LC-MS. But sometimes you might want to isolate a subpopulation of cells, or you want to analyze your biomarker in tissue, or you maybe don't want to measure a molecular entity directly, but for example, a protein function. And that adds a lot of more steps to the sample preparation procedure, and also introduces a lot of sources for errors. So you have to really look at that different steps in order to guarantee a reproducible and precise result. So we selected different case reports for you to show you how we implement such workflows.
So we want to kick off. With an example on microsampling. So traditionally, and everybody will speak about microsampling of dried blood. So traditionally, this has been used for measuring your drug of interest. But to my knowledge, it's not so extensively been used for biomarker characteristics. And we will be showing why this is a very interesting example, particularly when speaking about patient-centric sampling. This is really an intriguing component, Heike.
Yeah, so dried blood spot microsampling has been used for decades now with dried blood spots on paper cards. And we, as many others, developed a lot of assays using this microsampling technique. And we were always able to nicely validate these assays in the lab. But when it comes to study samples, then we always had really big issues. And I put some real life examples from past studies here. And you can see that, this was most due to inconsistencies in the blood sampling. Because when you do dried blood spot sampling, you have the problem that you can't add an internal standard from the beginning. So you can't compensate by an internal standard the recovery bias when extracting the blood from the card in this case. So we always try to convince customers interested in microsampling to not use dried blood spots on paper cards, but what is the alternative? And the alternative we like most is to use the Mitra devices from Neoterics. They have an absorptive tip, which can suck up a defined volume of blood or other biological fluid. And you can nicely see by eye when the tip is full, it's completely red. And this, we thought, offers less sources for errors in sampling. And I also put on the bottom a real-life example from a past study. So you can see this is more homogeneous, and we always gained quite nice results from that. And what is influencing the recovery from the tips or from any dried blood application, the most influencing factor is the hematocrit level of the blood. So if you have a high hematocrit level, the recovery might be hampered. And also if you store it for a longer time, then with high hematocrit samples, the recovery gets poorer. So the first thing we do or offer to our customers is a short test where we test the recovery with normal blood and with an artificially high hematocrit level. And I put an example here. That's a creatinine assay. Creatinine is a standard indicator for kidney function. And here we tested this in the middle with a normal blood sample and with a high hematocrit sample on the right side. And you see that the high hematocrit level doesn't influence the recovery from the tips. So we then used this to implement an assay, and we used the 10 microliter Mitra devices for that with a simple aqueous extraction. And we could really nicely implement the assay with a calibration range from 10 to 10,000 micromole per liter. And that also covers the endogenous level, which is roughly around 100 micromole per liter, and also leaves space for increased creatinine levels, which would indicate poor kidney function. And what is now the precision of such an assay? And we were really satisfied with this assay because the precision was really good in all calibrations and QC samples with a CV below 10%. And we were also able to demonstrate a long-term stability at room temperature for about 100 days. And when you store it frozen, then we could demonstrate stability for 259 days. So we were really happy with this assay, and would like to use that